So now after vesicular mechanism, vesicular trafficking, now let's talk about the lysosome. Okay. So in general, we know what lysosomes are. So they are the membrane enclosed organelles which will contain your acid hydrolases. So just, like in the previous lecture also we said, like the pH of your lysosome is less, right? It is around 3 to 5, whereas the pH of your cytoplasm is around 7. Okay. So if these lysosomal proteins or lysosomal enzymes are present in the cytoplasm, they are not active, right? They're inactive. Otherwise, what will happen? They will start degrading the material which is present in the cytoplasm. If it is like proteases or hydrolases, so all these enzymes can start degrading the other proteins. So since it is present in the neutral pH, they are inactive. Okay. Now they only get active once they enter the lysosomal lumen. Okay. So now lysosome function as the digestive system of the cell, serving both to degrade the material taken up from the outside of the cell and to digest the components of the cell itself sometimes. Now sometimes it is digesting its own components. For example, mitochondria ho gaya, chloroplast ho gaya. So that becomes the part of your autophagy. Okay. And autophagy is also known as PCD2. Programmed cell death 2, where apoptosis is known as PCD1. Please do remember, okay? Now, the lysosomal acid hydrolases, so they contain about 60 different degradative enzymes which is meant to degrade or hydrolyze proteins, DNA, RNA, polysaccharides, okay? अब इनमें से किसी एक में अगर mutation हो गया, that will result in the genetic disorder known as lysosomal storage disease. So this one is important. Okay, so most of these diseases which will result in the deficiency of your lysosomal enzymes. एक यहाँ पे मैंने दो एग्जाम्पल दिए हैं, but there is another chart where you can see there are many actually. Okay, so Gaucher disease, this is the common of all the disorders which results in the mutation of the gene which is encoding a lysosomal enzyme. Okay, required for glycolipids breakdown. So अगर ये enzyme आपका नहीं है और mutation is है, it results to Gaucher disease. Now coming to the eye cell disease. Eye cell disease is because the tag, the mannose-6-phosphate tag is missing from the enzymes, because of which these lysosomal enzymes are not getting targeted to the lysosome. Okay, and hence the lysosomal enzymes are not there, which result finally in the accumulation of the products and which is not getting degraded. Okay. Yeah, so this is that flow diagram which I was talking about. So here all the enzymes are mentioned. Okay, ये आपको याद करना है. अब कैसे करोगे? It's really on you guys, because कुछ भी पूछ सकते हैं, and all these have come once in the exam. So if your ganglioside GM2 is not having the enzyme hexosaminidase A, then you don't get GM3 and it causes Tay-Sachs disease. Okay. Now Gaucher, as we already mentioned, it is your glucocerebrosidase enzymes deficiency. Okay. Then from ceramide, if you're not getting to the glucocerebroside, it is because of your alpha galactosidase A enzyme. It is known as Fabry disease. Then sphingomyelin, you are not able to degrade into the ceramide. You know the ceramide में जब हम आप choline add करते तो आपको sphingomyelin मिलता है, right? Now sphingomyelin you are not able to degrade, because of which so much of sphingomyelin is getting accumulated, right? That is because of the enzyme sphingomyelinase is deficient. It leads to the disorder Niemann-Pick disorder. Okay, same way galactocerebroside is there. Now if suppose galactocerebrosidase enzyme is non-functioning, then it results into Krabbe disease. Now sulfatides are there which is not getting converted because galactocerebroside, then it is your aryl sulfatase A enzyme which results in the metachromatic leukodystrophy. ठीक है, ये अगर फ्लो चार्ट की तरह दिया है, सो दैट इट बिकम्स इजी टू रिमेम्बर. हर एक चीज को डिग्रेड करके सेरामाइड बनाना है, इतना आप समझ जाओ. ठीक है, सो यू नो सेरामाइड से स्पिंगोमाइलिन बनता है, सेरामाइड से ही हमारे ये gangliosides और glucocerebrosides, glycolipids बनते हैं. So just understand that everything needs to degrade and get back to the ceramide, the raw material of your phospholipids and the sphingomyelin synthesis. So when these enzymes are non-functioning, that results into these particular name of the lysosomal storage disorder. Yeah, can you do it guys? याद होगा? याद करना पड़ेगा. There is no option. Okay. Yeah. So I'll go ahead now. Now, so how the lysosomes are making their pH less. Okay. Okay. So as I was saying, like how this lysosome is maintaining is less pH. Okay. So if you remember guys, like when we started with the plasma membrane and the type of the transporters, we do talked about like V type ATPases, F type ATPases, P type ATPases, right? So this is the V type ATPases. So this is your H plus ATPases. So here what happens against the concentration gradient, okay, with the help of the ATP, the H plus ions are being taken inside. And that's how higher the concentration of H plus, lower the pH. Okay, so you just have to remember that this is your V type H plus ATPases, which is present on the lysosomal membrane, which is making sure that against the concentration gradient, all those H plus ions are taken inside. So the pH becomes less. And the moment the vesicle which is meant for the lysosome, they come and get fuses over here. Okay, what will happen? Those inactive enzymes will become then active. Okay, now this active enzymes, if suppose there are certain components which needs to be degraded, जो आपका बाहर से आ रहा है, endocytose होकर आ रहा है, so they go and get fused with the lysosome. Okay, so that becomes your phagosome. हमने जो पढ़ा है, right? Phagocytosis होता है, it goes and fuses with the lysosome for the degradation and becomes phagosome. Correct? Now suppose your one of your organelle is not functioning properly, let us say mitochondria, so that will be wrapped around by the ER membrane. So guys, here you have to understand one thing. Do I have the image? Okay, this is how the endocytosis and the lysosome formation is. Okay, we can come back over here later. Let me talk about the autophagy. So in the autophagy, if you see, lysosome is there. Let's say this mitochondria is not functioning properly. Okay, first, before it fuses with the lysosome, it needs to get wrapped by the ER membrane. Now, can anyone tell me why it is getting wrapped by the ER membrane? It does also have its membrane. Why can't it go and fuses directly with the lysosome? Is my question clear, Shruti? Question is clear?
Yes, ma'am.
Yeah. Always we say these plasma membrane, organelle ka jo bhi membrane hai, this cell membrane, they have got the similar composition of phospholipids. Then why can't mitochondria go directly and fuses with the lysosome? It's because the mitochondria and chloroplast, if you remember, they are the double membrane system, right? So total of 4 membrane will be present. Outer ka outer inner, inner membrane ka outer inner, correct? So they get trapped by the ER. Now this ER membrane is similar to your lysosomal membrane. क्यों? क्योंकि lysosome आपका ER membrane से ही बना है. So it becomes easy for ER to wrap all the unwanted components which is meant for the degradation so that it gets fuses with the lysosome. ठीक है? And the phospholipids which is present on the outer membrane of chloroplast and mitochondria varies because of its nature. So if you see later, we'll talk about that. So the phospholipids which is present on the mitochondria is little different as present on the other membranes. We'll come to that in the next session when we'll talk about the mitochondria. Okay, so the phospholipids are different as compared to the other membranes of ER, Golgi or lysosomes. So now what will happen? Lysosome ho gaya, aapka mitochondria aa gaya. It is like autophagosome, right? Because it itself is digesting its own component. Then it fuses to become the phagolysosome. And eventually it is degraded and all the amino acids, proteins, DNA, RNA, everything is broken down into their simpler components, released into the cytoplasm for the recycle. Okay, so I hope this is clear. Now let's go back to the lysosome formation, like how lysosomes are formed. So from here you can see ER hai, right? ER से वेसिकल्स बना और सॉरी ER से आपका गोल्जी में गया, गोल्जी से you do remember that those amino 6 phosphate and amino 6 phosphate receptor is there which is carrying all those acid hydrolysis, correct? Now they go and what happens is that here from here where acid hydrolysis are there, they go and bind to the endosome. ठीक है, अब एंडोसोम कहां से आ रहा है आपका? एंडोसोम इज कमिंग आफ्टर द एंडोसाइटोसिस. Okay, so these endocytosis will be having all these other membrane proteins and those cargo proteins are there, correct? So the receptors will be taken back and these components will be present. So this endosome, when your acid hydrolysis wala vesicle fuse hota hai, it results in the formation of the lysosome. Did you get it?
Ma'am, can you repeat once?
Okay. I'll go one by one. So I'll divide in 3 sections. Okay, so this is clear. Like from ER, the proteins are synthesized, it goes to the Golgi, and in the Golgi, it gets modified having the mannose 6 phosphate ka tag. Okay, so all the proteins which is meant for the lysosome, they let us say those are the acid hydrolysis having the mannose 6 phosphate ka tag bound to the mannose 6 phosphate receptor. Okay, this is first part. So the vesicle carrying the acid hydrolysis which is meant for the lysosome is ready. Now here already certain receptors are there. Okay, uska kya kaam hai? To go outside and fetch the cargo. Okay, the vesicles, or no, not the vesicles, sorry, to fetch the molecules or the components which is meant for the degradation, correct? So those goes and binds to these receptor. These receptor will endocytose. This is endocytic vesicle, correct? Now what happens? Eventually they keep on adding. ये एक नहीं होंगे, ऐसे बहुत सारे होंगे. They will come together. The receptors will get recycled so that it can go and fetch new cargo proteins. Okay. Now, when these particular proteins are there, this needs to be digested, right? For further use. Cell को nutrition चाहिए, वो कैसे करेगा? Once the acid hydrolysis containing vesicle fuses to that endosome. Okay, this is your matured one. Then the fusion of your late endosome with the acid hydrolysis results in the formation of lysosome. अब ये क्या करेगा? जो भी एसिड हाइड्रोलिसिस और जो प्रोटीएजेस रहेंगे वो जो भी कंपोनेंट्स लेकर आए हैं उसको डिग्रेड करेंगे और अपने सेलुलर फंक्शंस के लिए रिलीज करेंगे. Then again these receptors will go outside, they will express outside, they will bring the cargo again inside and इस तरह से आपका एंडोसोम बनेगा, लाइसोसोम में डाइजेस्ट होगा. Now it is clear?
Ma'am, there's one question. So lysosome ka pH is even more less is because of this acid hydrolase or?
No. Lysosome ka pH is less because of the H plus ATPase present. Okay. इसका काम है to take all those H plus from the cytoplasm inside it. Since less pH makes your acid hydrolysis active. वो किस लिए? Why they have arranged like this? Why the nature has made these arrangements? Because just imagine on the way accident हो गया. What happens? The cargo जो है वो सारा आपका spill over हो जाएगा, बाहर गिरेगा, right? And if just imagine those acids are getting spill over everything, acid के सामने कुछ भी आएगा तो it is gonna damage those also. Same way if acid hydrolysis were active at the neutral pH और ये vesicle आपका टूट गया by mistake because of certain accidents happened. Okay, so acid hydrolysis will come in contact with the proteins which is present in the cytoplasm doing their own function, correct? So wherever this acid hydrolysis or other proteases will come, they will start degrading all this, which I don't want to happen, right? So nature has selected these acid hydrolysis to be active only at low pH. So neutral pH में अगर by chance accident होता भी है, so they are just meant for the degradation then, क्योंकि उसका कुछ काम नहीं है वहां पे. Otherwise, if they are reaching their target organelle correctly, So inside where the pH is less, they will get active and they start degrading all those components जो हम बाहर से लेकर आए हैं खाना to digest.
Okay.
Just imagine as if you eat something, your acids, your enzymes are all digestive enzymes are already present in your stomach, right? So जब भूख लगती है, that gurgling sound comes. That is nothing, just the decrease in the pH, which is making your enzymes active. Now you start eating something, you feel relaxed. In the same way, a cell is also bringing something from outside to eat. तो लाइसोसोम क्या हो गया, उसका डाइजेस्टिव सिस्टम हो गया, जहां पे उसने डाला एंजाइम्स एक्टिव हो गए एंड दे स्टार्टेड गेटिंग ऑल दोस रिसोर्सेज, एमिनो एसिड्स, डीएनए, आरएनए, व्हाटएवर इट इज प्रेजेंट, जो भी वो बाहर से खाना लाया है, ठीक है. दिस इज हाउ दे गेट देयर डीएनटीपीस. नहीं तो ये नया डीएनटीपीस, नया आरएनए, नया एमिनो एसिड कहां से आएगा टू सिंथेसाइज न्यू थिंग्स, राइट? दे नीड रिसोर्सेज. So this is how they get their resources from outside. Now it's clear?
Yes, ma'am.
Yeah. Okay, so that's all, guys.
So that means at late endosome level they are zymogen form, and only after they get
Yes.
into lysosome, that's when they become active.
Yes. But don't use this zymogen in ye sab, because those are meant for the stomach enzymes. Okay?
Okay.
Yeah, but the concept is that only. Good. Anything else? So I'll be stopping here, and 1 second. Let me stop the recording.